Ready to Emerald Ash Borer Awareness Week 2024. We are celebrating this week with a series of 6 Don't Move Firewood led presentations that will be taking place over the course of 3 days. The Don't Move Firewood campaign is led and run by the Nature Conservancy with financial support from the USDA APHIS and also coming up soon, US Forest Service. For day 1, we will have 2 different webinars. The first is this one that you are on right now, which is Ash Tree Breeding and Resistance to Emerald Ash Borer with Jennifer Koch and Rachel Kappler, and they'll be speaking in a moment. The second one is at 5 o'clock Eastern, so a little late for you Easterners, but during the workday for everybody else, so that our Australian researcher doesn't have to give a presentation at 3 in the morning. And so that one will also be fascinating, and I urge you to log in. Laurel will be putting in the chat the link for the full series if you'd like to register for additional webinars beyond the one today. Each link is unique to the webinar. We will be doing about 40 minutes of presentation and then about 15 minutes of Q&A, depending on how that goes on in terms of timing. Please use the Q&A box feature, not the chat, for questions for the presenters. It's much easier to keep things separated out like that. And then all the presentations will be recorded and posted onto the Don't Move Firewood YouTube channel. Certainly by mid next week, we will get them all posted, if not sooner. So with that, I am going to turn off my screen share and hand over screen share to Jennifer Koch with the Forest Service. Jennifer, you should be able to share your PowerPoint now. That's looking good. And just click on presentation mode when you're ready, and you're ready to go. That looks great.
Okay, so it's looking good.
Yep, and you sound good too, so go ahead.
Okay. Well, I'm Jennifer Koch with the Forest Service Northern Research Station in Delaware, Ohio. And I'm really excited to be kicking off EAB Awareness Week. And thanks everyone for joining us. I want to start with acknowledging my Forest Service colleagues who have helped with our breeding and resistance research to emerald ash borer. Kathleen Knight, Dave Carey, Mary Mason, Therese Pullen, and Toby Patrice. And then Rachel Kappler, who is the coordinator of the Great Lakes Basin Forest Health Collaborative at Holden Forest Garden, will be picking up after me. So to start, okay. No, it doesn't want to let me go forward.
Sometimes you just have to click once on the slide and then click forward.
Oh, okay. I thought, how do I hide the bar at the top so I can see my slide? Or you can't do that?
Sometimes you can drag it to the bottom so it's less annoying, but
yeah, it keeps moving. Well, it doesn't go. Okay, it's fine. Anyway, I wanted to, I'm sure all of you are aware of the importance of ash, but sometimes, depending on where you're at, all of the ash is gone. You don't think about it. So I thought it was a good idea to just review some of the really important things that ash does and has been involved in it's sort of part of American life. It's been used for iconic American products like the Louisville Slugger baseball bats, no longer though. Fender and Taylor guitars still both use ash wood to make their guitars. White ash is an important timber species. Both green and white ash are common landscape and street trees. Should point out Keith Richards down there from the Rolling Stones is holding a guitar made with ash wood. So EAB has really impacted the economics, wood products industry, as well as the nursery industry. In addition, ash are very important ecological species, especially in riparian and wetland forests. Green, black, pumpkin, and Oregon ash all play a role. One really important example is the regulation of wetland hydrology, especially with black ash. In the state of Minnesota alone, there is a risk of conversion of over 300 million acres of black ash dominated forest from forested land to non-forested land. And it's thought to potentially be a permanent conversion which could contribute to climate change in a day and age when we are trying to plant 1 trillion trees to help mitigate climate change impacts. You may not be aware that agroforestry is another important role for ash. It serves as a riparian buffer. You can see my pointer, but you can see here is ash around this river. Up in the corner, there is farmland. So this acts as a buffer and keeps pollutants out. So as we lose our ash, you're going to see an increase in non-point sources of pollution. Okay, and the reason we're here today, we all know the threat is emerald ash borer. This is just updating everyone where we are at. How bad is it? So the red dots on this map are showing all of the locations where EAB has been detected. It is now 40% of the range of ash that's infested by EAB. 9 out of 16 of the US species are included in this range. And according to the International Union of Conservation of Nature's Red List, 5 of our species have now been named critically endangered for potential extinction. That includes green, white, black, blue, and pumpkin ash. Carolina ash, farther down south, is listed as endangered, and Oregon ash, which is over on the West Coast, a couple of years ago, EAB made that jump, and so that is now considered near endangered. But I'm here to talk about hope. I'm the person that gets to give everyone some hope. And I wanted to start with the story of lingering ash, which all started with ash monitoring plots. And I put up this publication. You can find it on the Forest Service Tree Search website. If you search tree search, you should be able to find it. Kathleen Knight is the first author. Rachel Kappler, who is speaking, also was heavily involved in this work. So basically, what this is showing is a series of monitoring plots that Kathleen installed, and she and her crew and collaborators monitored these from 2005 to 2015. Over 3,000 ash trees were tracked. Other trees were tracked too. It included 5 different ash species in and amongst these plots, and they intentionally targeted different ranges of ash densities and habitats. Because being ecologists, they were interested in looking for factors that might play a role in the trajectory of EAB killing the ash trees. At the same time, they were monitoring EAB population dynamics, and that's shown in this slide here. You can see the purple prism trap hanging in an ash tree. And here's a close-up showing you an emerald ash borer. So a couple times during the season, those are taken down and the EAB can be counted, so we can compare the population from one site to another. Among all of Kathleen's plots, she saw the same trajectory. Eventually, all of the mature ash, she was measuring as mostly trees greater than 10 centimeters in diameter. The only difference between plots was the amount of time to mortality. So it was from 3 to 7 years from the time that the first symptom of EAB was observed. So in this diagram, the green represents healthy ash and the purple represents the EAB population as measured by those traps that I just showed you. So this starts out being a very healthy plot with healthy ash trees. EAB enters and we start to see some decline. EAB population increases and there's a very sudden decline. Shortly following that decline, the emerald ash borer population decreases. And all of the ash trees that were being monitored have now died. The EAB population continues to decline. I will say it doesn't ever quite go away though. She still continues to pick it up on her traps. But the great news was, and the surprising news was, not all of the trees followed that model. When they were hiking into sites to measure or collect data on their plots, they would find a healthy tree, or at a subset of plots, they actually found healthy trees when all of the other trees had died. We coined the term lingering ash because we didn't want to be overly optimistic until we had some data to support it. We didn't want to call the trees resistant. As that turns out, not all lingering ash trees are resistant. And Rachel might mention that in the latter part of this talk. So we've been finding lingering ash trees both individually and in clusters. We don't think that they are escapes because we often see them like in this photo where there's a dead tree that was killed by EAB right next to a very healthy tree. On the far left is a stand in Ohio at Alum Creek State Park, showing you all of the downed dead ash trees. In this area was long dead, but just 2 miles away, we found 2 really big, healthy ash trees. In addition, Kathleen and her crew found a larger cluster of lingering ash at Swan Creek floodplain. This is this way in this area of Oak Openings Park near Toledo, Ohio. They found healthy ash trees there, so decided to do a deeper monitoring of this site. I think they covered about 2 miles along the floodplain. And you can see the downed ash here. From that data, they estimated 11,000 trees had died in this area. But they found plenty of live ash trees. 284 were alive, 108 were alive and had healthy canopies. So that's 1% out of all the trees they looked at in this area. Since 2010, there's been continued monitoring at Swan Creek and other plots. And the general trend seems to be that trees with healthy canopies are surviving over time. Not all of them, but many of them. Declining trees tend to go on to die, and even healthy trees are weakened. So we sometimes see them dying from other causes. And that's shown in this picture here, where this lingering ash was blown over in a windstorm. EAB are persisting at low levels. That's being detected through monitoring of the EAB traps, so they can continue to attack these healthy lingering ash, as well as any regeneration in places that it does occur. It doesn't occur everywhere. Oops. So is it possible that these lingering ash really have resistance to EAB? I think I already gave that away, but anyway, as a geneticist, this is something we were really interested in. And there was a lot of pushback. People just didn't believe it was true. They didn't think that if a tree species didn't co-evolve with the insect, that there could not be resistance. So I began to understand that part of the problem was people don't understand what we expect when we're talking about resistance. What does it really look like? And it can be partial resistance. And partial resistance may simply allow trees to live longer than susceptible trees. But the good news is they can be further improved through breeding. So what we see is exactly what we would expect from a genetics perspective. Because they did not co-evolve, it would not be selected for, it would be no advantage to have any resistance. So the most common is going to be our highly susceptible trees. So most of them are the ones that are just totally hammered like this. But the lingering ash, most of them the vast majority of the ones that we see would look like something that has partial resistance, meaning they are being attacked, but it's healing over and the canopy is remaining healthy. So we are not looking for immunity. We didn't expect that. However, in a couple of trees, 2 to be exact, we have found trees that we couldn't find any evidence of EAB attack. So the criteria of selection lingering ash for resistance testing, we looked in natural forests long infested by EAB. The trees had to be large enough during the peak EAB part of the outbreak because EAB have no well-known size preferences. And we were looking for a healthy canopy, and we want trees that are alive for at least 2 years after the mortality rate levels off. Remember the graph that I just showed you. And I think Rachel will review that again later. Just to give you an idea, a winter photo of a healthy lingering ash is here on the left side. You can see a full crown, and you can see the fine twigs are intact. Kathleen ranked the trees, the health of the canopy, on a scale of 1 to 5, with 1 being healthy and 5 being dead. When we're looking for lingering ash, we are hoping to get 1s. Sometimes we'll get 2s, but we have the best luck with the healthy ones. And just to go over a review of the general process of resistance breeding, this slide's adapted from my colleague Leela Wilson. The basic steps are you collect seed or scion from your healthy ash trees that are in natural forests, put them in the ground and field test by just allowing EAB to infest them. And then you select the best performers and bring them all together. So when you put the best trees together, you make a seed orchard that's going to produce better seed. And that seed can then be used for restoration. And the goal of restoration is to get a self-sustaining population that can continue to evolve and increase resistance. In breeding programs, though, we also like to add this other step of testing for resistance using potted plants, either younger seedlings or freshly grafted grafts right from the lingering ash in the woods, because this can help reduce the number of trees that we put into the field test. And the field tests take a lot of land, a lot of labor, a lot of time, and it's very expensive. So if we can screen out the worst performing trees and not have to get them in the field, that saves us some time and money. So the bioassay, or the test for resistance that we are using, we work with entomologist colleagues, Therese and Toby, who rear adults, get them to lay eggs. They actually get them to lay eggs on coffee filters through a screen. They ship them to us on coffee filters, and we cut out individual eggs and tape them to the tree. 8 weeks later, we go back, we find the entry hole where the larvae entered the tree, and we very carefully trace the gallery until we find the larvae. This one is a nice healthy larvae. And over here, you can see a smaller larvae that died. The gallery is very small. It died from host defense responses. This one is showing the entry point of the larvae. It didn't even make it all the way into the tree before it died. So the ones we're going to select to go into these field plantings will be these ones that are showing host-killed larvae. So better trees are those that are killing more larvae. On this slide, and breeding increases the amount of resistance. And this slide is data based on the bioassays. It's confusing, but I want to try to simplify it for you because it does make an important point. So we're looking at individual seedlings from each of these families. This one circled in black is a susceptible seed lot. And this is the proportion of larvae that were killed by each dot represents a different seedling. This line here at 0.12, that's the best performing of the unselected or susceptible seedlings. So about 12% of larvae at best were killed. But when we looked at our best performing lingering green ash selected straight from the natural woods, after we watched over 95% of the trees die, the best tree we found killed about 45% of the larvae. So our lingering parents are in between about 20% to 45% of killed larvae. But if you think about it, even if 1 out of 4 larvae is killed, that's 25% less damage to the tree. And that would be enough to allow that tree to live longer. So once we bred the lingering ash, so use 2 parents to cross them and produce seedling families, 80% of those families that we have tested have produced seedlings that are at or above 45% of the larvae being killed. 40% of our families have seedlings that are at or above 80% killed. We actually have families that are producing larvae that kill 100, or trees that kill 100% of the larvae. And this is in the range of the Asian species Manchurian ash that did co-evolve with EAB. So we're very excited about the results. I do want to point out, though, that the seed produced from the seed orchards, we aren't looking for 100% immunity. We do expect the seed to be diverse. We're looking for an increase in resistance in the frequency and the level in the population. We want enough survival so that they can survive. And a key part of this is to maintain genetic diversity. So we want to maintain diversity in the resistance mechanisms, so it's not going to be easily overcome by the EAB, and that the tree population can retain resilience to adapt to changing climate, other pests and pathogens, and maintain their adaptive traits. As I said from the beginning, we have to always test in the field. One of the reasons we want to test in the field is to make sure our bioassay is indicative of field performance. So far, that's looking good, at least at the species level. And we also want to assess environmental impacts on resistance. Because when you're doing the bioassays, they're in a very controlled environment, and you can put a really resistant tree, but if you put it at a site where it's not happy, that can have a negative impact on the level of resistance that it displays in the field. So this picture here, I think was actually taken by Rachel. It's at Holden Arboretum. That's the parents that were selected straight from the woods. And over here in Delaware, Ohio, these are progeny from the crosses that we've done that we're testing out in the field. And I wanted to take a minute just to talk about heritability, because this is the foundation of breeding, and this is the reason why breeding works. And when we breed trees together, we can get improvement. It's complicated, but yet it's also somewhat intuitive. It's basically thinking about how much do children, or when we talk about trees, progeny, resemble their parents. So, for example, if these are each lingering ash parents and they have a lot of children, you don't expect all of your children to look just like you. It depends on what genes they get. So in this case, this progeny seedling didn't get any of the good genes. Each of these got some of the genes, so they look a lot like their parents. This one got the best genes from both, so it looks great. Heritability is measured on a scale of 0 to 1, 0 being the highest. So if we think in terms, again, just of humans, as parents, you may see a trend that your children are closely related to you in your height, or the average between the two parents. Look, so that it's either like one parent or the other, or the average between the two. And human height has a heritability of 0.8. Our green ash seedlings, when we looked at over 770 progeny from 27 different families, we're at a heritability of 0.63 to 0.87. So these numbers may be complicated, but the punchline is they're high enough that tell us that breeding will work. And breeding additional generations, we expect to get additional improvement in our populations that we're producing. So at this point, the strategy we're taking is doing crosses between as many different lingering ash parents as we can, selecting the best progeny from each, and putting all the best trees together in the seed orchard. Now, the challenge is that all of the trees that we have collected so far are in this little circle. So mostly in Ohio, Michigan, part of Indiana. So it's not appropriate for the seed produced from here to be used to restore ash all the way down here, say in Alabama or Georgia. It's not going to survive. It's not adapted to grow in that climate. So this process must be repeated regionally using new genotypes in order to be able to have resistant trees appropriate to grow across the entire range of green ash that's shown in this map. And that's going to require a lot of help. But we are on the road to saving our ash resources, because breeding is an important part of integrated pest management. As you increase host resistance, you're reducing habitat for the emerald ash borer, and hopefully biocontrol is keeping the EAB populations down, and we're headed towards that sustainable ash resource in North America. And the next question is, how can you help? And so at this point, I'm going to turn it over to Rachel.
Thanks so much. Hopefully everybody can hear me all right. And what we're doing is taking a look at a couple of different resources that are available to people that if you have a moment to take a screenshot or write it down, or I'll put it up later. We have a couple of different databases that our lingering ash trees can be reported to, although I'm also helping with coordinating that, so you can always just email me as well. And one of those is TreeSnap, which is an app that is I'm going to cover a couple of different tree species. I'll go over that in a minute. Each of these I'll go over here, such as what we call our MAMA program. It's monitoring and managing ash, and that helps us a lot with both the before and after times for looking for lingering ash. And what I do is work as the coordinator for the Great Lakes Basin Forest Health Collaborative. And I'll be going over a little bit more in detail on that. But basically, the main focus of this Forest Health Collaborative is to make sure that the partners who are interested in doing more for pest and disease resistance breeding are coordinated and getting all the training and resources that they need in order to help out with their current and future projects. So if you go to the next slide, I'll get into a little bit more of each of these. But first, don't forget that what we're looking for is a lingering ash that has a certain criteria in order to be reported on either to myself or to TreeSnap. And just as a refresher, we have areas that we're looking for them in as a natural wooded area where more than 95% of or most of the mature ash trees that you know about have already reached mortality stage from emerald ash borer. We see that there's a little bit of a variation in this due to the different latitudes that people are living at. So it could also be maybe 4 years since 50% of the mortality has been reached in ash trees in that area. So there's a little bit of wiggle room depending on how things going. And usually we ask to take a look at that tree for a couple of years to see how it does. Large enough, of course, to be preferred by EAB during the peak infestation that happened at that location, and with a healthy canopy during the leaf out months between June and September. Now, again, we love having all the reports of lingering ash trees. Not all of them are going to be 100% resistant. And that will be one of the reasons why we'll usually take another year to look at the lingering ash tree report again and look at the tree again to see how it's been doing, excuse me. I'll go ahead and go to the next slide. And just as a reminder that this is what it had looked like. You saw a graph like this before. For the southern Michigan, northern Toledo area, on the green line for the ash that are alive, and the purple line for the number of EAB per trap that had been put out. And with that, you'll see that there was a time where, we knew we had EAB in the area, but we weren't going to be able to tell which trees were lingering ash trees. So there was a waiting period that needed to happen in order for us to know for sure that we had trees that were making it past a critical stage, a sort of a natural test from EAB. Next slide, please. So the TreeSnap app is something that comes in very handy, not for just for us, but for lots of other tree researchers as well. So this is some screenshots of the application, and it also has a web page that you can go to log in to see the map and things, but you'll need the app in order to be at the location to enter the tree. And so if you're interested in helping out with other projects, these are not all pest and disease resistant related, but is something that is useful on different projects. I have access and am able to verify the reports that are put into here. And a lot of these data points that you need to enter in for that, it can have some explanations within the app to help you input them so that it's not as difficult as it seems at first. Next slide, please. And then with the Ecological Research Institute, which is based in New York, they have a Managing and Monitoring Ash Projects program, and we call that the MAMA program. Monitoringash.org is their webpage, and with this, they have come up with a couple of different projects and procedures that go along with them in order for us to help look at what's happening in a certain location with their ash trees and EAB pressures. And that can be also done in order to assess whether or not you have EAB in the area, assess how the trees are reacting to EAB over time, and know when it's time to start looking in that area for lingering ash trees. So each of these have their own little section of a different phone app called ANIC Data, and that allows them to help work with their partners in order to have data available to that between them and their partners. They are able to assist professionals or groups of citizen scientists who want to start with one of these different projects, and it will all depend on what stage of EAB pressures you're in. Please go to the next slide. And so they also have come up with this lovely table that can help you figure out where things are at. If you don't know where your state is at for its EAB pressure, there's emeraldashborer.info, and they'll have like the year that your county first got EAB detection. So with the pre-infestation, you can start to assess where your ash trees are and know where they are. Early infestation, you'll have some EAB being trapped. You'll have some trees showing some declining canopy health. And mid-infestation, you'll have it more mortality happening to those trees. And so usually we have the situation where a lot of different management decisions can be made based on what stage you're in, but also take into note that being able to monitor the ash trees through any of these stages is helpful for us to know where the threshold is for when we should look for lingering ash trees. And the sooner we know for sure that those trees have been watched and that mortality level has been reached, the sooner we can go out and find those rare trees that we're looking for. But when we're in the late stage of infestation and a lot of them are dead, we'll look for lingering trees. But we also have to keep an eye out for making sure that we're not taking in a tree that, got a lot of light and a lot of water and had a good environmental conditions and is actually the next generation tree growing up quicker than average. Next slide, please. So with this, you can go to their website. You can work directly with these people from the Institute. They'll help with training you on how to do any of these different projects. And they're based out of that location, but they've also have trained myself as well. So I could also assist you in that as well if there's any groups or locations that a project might want it to be started. A lot of the mortality data from the Mama Citizen Science Project really helps us with understanding how EAB is spreading and causing an impact on the forests. And we've noticed more and more that is not reacting, of course, the same way as it did in the Toledo, Detroit area. And you end up having a little bit more of pockets occurring in how quickly and how determined the EAB are in that area to hit a complete mortality of ash in that pocket. Next slide, please. So with this, so far, Mama has been able to look at certain locations in New York and being able to see those patterns, are able to understand better what we're going to be looking for, but are also finding lingering trees of these different species. So we're really hopeful that this is going to be something that can spread to other states where they're looking to collect more data on their location. So we have the next steps occurring for future resistance breeding in many different states. Next slide, please. And so you can see here, you can find this on their monitoringash.org page, but you can also search for it at anicdata.org. But I would definitely encourage, if you're looking to start one of these, to reach out to outreach at monitoringash.org so that they know that you want to start one of these, and they'll be on the lookout for any help that you might need, or to be able to at least know that something has been started. And so with that, we have the ASH EAB surveys to know if EAB is in the area. monitoring plot networks that helps out with the long-term year-to-year tracking of a location to see when we could look for lingering ash, which then leads into the separate section of the project where you would enter lingering ash tree data. But they've also come up with a new rapid ash mortality assessment. This one here is going to be a bit more helpful when we're working with people who may not have the year-to-year commitment, or might have a situation where we think it might be time to start looking for lingering trees, but we don't know for sure. So let's just take a check and see where we would go from there. Next slide, please. Now, with my coordination of the Great Lakes Basin Forest Health Collaborative, I'm stationed out of Holden Forest Gardens, which is near Cleveland, Ohio. But I work throughout the Great Lakes basins and those states of the Great Lakes. And we're not just focused on emerald ash borer and the resistance to EAB, but these other pests and diseases are on our radar as well, and are where we're trying to put some time and resources into either coordinating researchers that need to have more things happening with them, or coordinating management or citizens where we might need some more on the ground location or actions happening with them. This will be working out with training and partnerships through people, and it can be not just organizations, but individuals as well, and individual private landowners even. Next slide, please. With that, that means we have all of this information of who has what resources available in order to help out with the many different tasks and projects that go along with resistance breeding. And I help with finding new people, keeping them up to date on what activities are happening, connecting them with others that they might not to network with. And with that, if any information is needed, I'm here to help with either virtual or on the ground monitoring, or on the ground training, so that they understand the different, whether that's the monitoring stage for looking for lingering ash, or whether we get into the further on propagating, growing, and breeding stages of the program. Next slide. So in summary, we have a lot of EAB things happening across North America. It is a severe threat. It is attacking the many different ash species that we have on our continent, but monitoring is allowing us to observe what's happening naturally and being able to amplify that a little bit quicker than it would occur naturally, with us being able to identify lingering ash and being able to have that testing occur and monitoring happen afterwards to see how well we can get new seeds available, growing with their ability to work with the Forest Service and other researchers. These methods are helping us identify and propagate trees, and we have our data showing that this breeding process is working for us. Now, where we're at right now, we're needing partners, of course, to help in all aspects of establishing our resistance breeding program. Currently, if you're in the Midwest area, we're looking for properties that would be able to establish a planting of ash trees in a sort of an orchard-like manner with a deer fence. So if there's any interest in that, please let me know. I'll give you a little bit more details on how that is going, but it allows us to give us more property to have more field tests for these individuals, not only of the next generation that the Forest Service is breeding, but of new trees that people are finding in new locations where we might want to conserve copies of those natural lingering ash trees for the long term. Next slide. So thanks for your attention. That's how you'll get a hold of both of us, and I'll let Jen add any summary remarks that she might want to put in there as well before we go to questions.
Thanks, Rachel. I didn't really have any summary remarks other than what you just talked about, except to encourage people to get involved and to reach out to one of the resources that Rachel described, or contact either one of us with any questions about anything that you might be interested in getting involved in. And of course, it's always important to acknowledge the people who give us the funding to do what we do. So take a look at that. We've had a lot of support from the states, from the Forest Service, APHIS, the Nature Conservancy, and Great Lakes Restoration Initiative, which is funding the Great Lakes Basin Forest Health Collaborative. So I think if we have time now, we should have time for questions.
Yeah, we do. You guys were extremely timely, so I really appreciate that. We have 3 very good questions in the chat right now, so I will read them off from the top because I know not all participants are able to see the Q&A. Sorry, 3 really good questions in the Q&A, not in the chat. So number 1, we have an attendee who asks, how do the ash trees kill the emerald ash borer larvae? So the mechanism. And then they say just healing over the boring holes or tunnels.
We don't, I mean, it does appear that way because when we find them, they're usually encased in callus tissue, which is a wound response that trees have. But what triggers some trees to actually do that and recognize that the EAB is there or not, we don't know that mechanism, and we also don't know if that callousing is happening after the EAB has already died. So we really have no idea what the true mechanism is. If something in that tree is actually toxic to it, or if it's physically just being healed over faster than the EAB can outgrow it. But the important thing with breeding is we don't really have to understand the exact mechanisms. We just need to be able to measure resistance and be able to measure an improvement with every generation of breeding and selection. We just have to be able to select the trees that do have resistance.
And Jennifer, when you see that response in the Manchurian ash that co-evolved with emerald ash borer, does it physically look the same?
Yes. I used to have a slide that showed comparison with multiple different Asian species and compared to multiple North American lingering ash. And there's, I showed 2 different examples of what, like the one where it had a short gallery and was calloused over, and one where you could tell it was just a hole where the larvae started to chew in and didn't even make it in. We see that in the Asian species as well. It's very similar.
Okay, great. Yeah. Gary Fish from Maine has a question that we partially touched on in the subsequent part of the presentation, but actually I'd love you both to dig into the answer, which is, are you also working on white ash resistance? Because your introductory examples were focused on green.
Yes, we are. We're working on green, white, blue, black, and Oregon ash. However, we have the most data on green ash, so that's why I focus on that. We're catching up with white ash, so we'll have heritability soon with that. Black ash, we are happy that we just got funding to hire a second Great Lakes Basin coordinator who will be located up in Wisconsin. And when that happens, they'll be in a better place closer to black ash to hopefully start finding lingering ash and testing even seed lots. Although we have we do have some lingering black ash that we've already obtained, and I believe ERI and Cornell have identified some. So we're starting to get caught up with black ash.
Great. One quick note for those of you in Maine, like Gary, sometimes black ash is also called brown ash. That's both Fraxinus nigra. It's the same species. So if you're more familiar with the term brown ash, it's the same tree as black ash.
Yes.
Okay, our last question in the Q&A, but we absolutely have plenty of time for more, is what kind of rootstock is being used for the scions, and could that rootstock have an influence on the level of resistance that you see?
Yeah, that's a good question. And that was something that we were curious about from the very beginning. So we actually did some reciprocal grafting, and we didn't see any differences. There are some papers that show there's differences in phenolics, but as far as our bioassays, it doesn't make a difference. So we use green ash because it's the easiest to germinate and the quickest to grow of the species. And the horticultural cultivars of Manchurian ash, Mancana, that's typically grafted onto green ash and has been the whole time. So there's a lot of data showing it doesn't have an impact on the resistance of that. Grafting in and of itself kind of reduces the impact. It's a stress. So grafts usually are a little bit more susceptible than seedlings, but it doesn't take a tree like that is highly resistant and make it susceptible. It just may kill a few less larvae.
Great. And the advantage to grafting, of course, is that you're effectively cloning the parent, whereas if you had to wait for seed, that would be a child of the parent, which is an entirely different tree.
Yeah. And the other thing that we're doing, the other problem with grafting is that the rootstock is susceptible. So that means it can still be attacked by EAB. So we have worked out a protocol to do rooted cuttings that works really well in green ash and white ash. We've gotten it to work in Oregon ash and pumpkin ash, but black ash has been a real struggle. We're still trying to optimize that and get that to work. The rooted cuttings are great because then you have the exact genotype. You can make copies of it on its own roots. So you can use that to put into your seed orchards. You can use that to have replicates for experiments without having to worry about any influence of the rootstock or the susceptibility of the rootstock.
Yeah, that's great. So we do have 10 more minutes available. Those were great questions. At this point, if anybody wants to put questions in the chat or the Q&A, that's fine. Or I believe if you want to raise your hand and ask a verbal question, we can authorize that as hosts as well. So if anyone else has a question. Oh, look at that. Somebody actually did it. Laurel, do you know if we can authorize that person? Of course, I immediately lost track of them. I'm sorry. Raise your hand again, whoever that was. There we go. McKay. Let's see if I can do that. We're really in the future over here trying to figure this out. All right, Laurel, you figure that out and we will, or try to, and we will address another one of Gary's good questions, which is, Gary asks, when do you start looking for lingering ash? How many years past the first discovery of, I would presume, a local infestation?
Are you able? Oh, go ahead.
Oh, go ahead. You can answer, Rachel.
Okay, so usually when we were looking at the mortality that had occurred in the main zone near Detroit and Toledo areas of Michigan and Ohio, our graph had shown, it had been about 6 years on average for the locations to have that really high or complete mortality of ash in the surrounding area. But as the pest had moved to different locations, we've been noticing that it can take longer or a shorter period of time depending on that local area. So that's one of the reasons why we like to make sure that what I usually do is make sure I understand what the original date of finding an EAB in the county was, so that I can sort of have a guesstimate of how long it's been in the area. And then also having some field data in order to know how the forest trees have been doing. And that can be either anecdotally from foresters who are working in the area, or it can be from forest inventory analysis data, or from us putting out those monitoring plots in order for us to assess where we're at with that. But I usually am, it's gonna depend on the spot, unfortunately, but because it's a faster moving eating beetle than say other kinds of pests or funguses that can take longer. This one tends, I would say, on that average of somewhere between 6 to 10. It might be up to maybe 12 years before we start to see in any location that high mortality of ash. But again, the number of years varies based on the location and the local ash population density and things like that.
Okay, fantastic. We're gonna experiment and see if Mackie or McKay Burley can speak, because we're gonna try it. So please go ahead and take yourself off mute if you can and ask your question. If that works, we will find out.
Hey everybody, can you hear me?
Yes.
Yes.
My service is a little choppy, I'm so sorry. So I work for the St. Regis Mohawk Tribe. I'm their plant nursery technician, and we recently did a collection of black ash. Well, I guess not recently, it's been a couple years now. And we stored the seeds away, and I don't want to be storing them away in like short-term storage for much longer, and I'm feeling a little overwhelmed on like what my next steps should be. So I was wondering if you folks had is it worth it to use these seeds if they're not lingering ash? Should we be starting an orchard? I'm just curious of what your opinions would be if you were in my situation.
That's a
So,
great question. Yeah.
yeah, it is a great question. And I didn't really have time to go into the various species. Black ash, we have a small bit of data. It's from 5 seed lots that were collected before EAB attacked an area. And we just wanted to know, because there was not very much data at the time. So we screened these 5 seed lots, and we found that we could pick up a good amount of trees that were able to kill more larvae just from screening straight seed lots. About 1 in 25 versus 1 in 1,000, or over 1 in 1,000. But then the problem was black ash, you may be aware, or brown ash, it takes less larvae to kill black ash. So we're not sure that, well, our bioassay, we did it 2 ways. We did the bioassay at 8 weeks, and we also did it in potted plants that we left for a full year. So we did get better survival even in the potted plants. So I guess my short answer is there's something, black ash seems to recognize EAB and mount a defense response, but sometimes that defense response ends up being detrimental to the tree. And I think that's why it sometimes takes less larvae to kill black ash. But yet we're seeing a higher frequency of the ability to recognize and kill larvae. So it may be worth putting out progeny test plantings if you have the resources to do longer term seed storage. I don't know how many years. I know our seed storage facility has viable seed after 8-10 years. So if you want to save some of it and try and plant some of it, that might not be a bad idea.
And I'm not quite sure what the paperwork is like, but for our seeds that we're keeping for long-term seed storage, it's going to the National Seed Labs long-term storage facility. But I'm not quite sure if there's rules and regulations and processes and paperwork on it between them and tribal nations.
That's an interesting idea, Rachel. So he could investigate whether or not putting him into the National Seed Storage Lab was a good longer term solution, just given a lack of other sort of obvious plan.
Mm-hmm. Right.
Yeah. So for example, when we store our seed with them, it's marked so that it's only available for research, for example. So they may be able to store it and not distribute it just to anybody.
Sure, so market for use by the tribal nation or by permission only or some other mechanism.
Correct. And then there's also the NCRS that in Iowa has long-term storage, and they might be another resource.
This actually gets to the last question. And also, McKay, if you want to reach out to Jennifer or Rachel, their emails are here and you can pursue this conversation and some good ideas in the future from that way. But we have 2 more minutes and I wanted to get to the last question I see on the Q&A, which is from Connor Hagemeyer. And that is, what are the plans or processes for making this more resistant plant material available to other researchers and breeders or for reforestation projects? It's a big question for 2 minutes.
Yeah, we are working with a lot of other researchers. I would say, I don't know, 4 or 5 different universities. Ecological Research Institute, Holden Arboretum, Morton Arboretum. So we are making some materials available for other researchers for different projects to answer different questions. Most are revolving around trying to make this screening process quicker to accelerate breeding, and also figuring out like how far we can move things when we go to plant them. But as far as other breeders, through the programs Rachel described, they could get involved and potentially start propagating material. Depends on where they're at, what's appropriate. If we have materials for their area that would be appropriate. If not, they would have to start at the stage of helping find lingering ash that would be appropriate for that area. We're a ways out for restoration projects. The first restoration seed that will be available is going to be appropriate for Ohio, Michigan, maybe part of Pennsylvania and Indiana. And we still need to add more parents to that. And we do have seed in hand that we plan on planting a progeny test. It's open pollinated seed that we collected like 2 years ago now. We're doing germination tests this year, but it was so long after peak EAB that these were lingering ash parents. So we were making the hypothesis is that the pollen parents were also lingering ash. So there might be some natural improvement in these seed lots. So that's going to be the first sort of outplanting that we'll be doing. So I think I answered the best. If not, email me.
Yeah, I will add that I saw in the earlier chat that Connor is located in North Dakota. So it's a good example of how that would not currently be a location where it would be, likely that the trees should be moved from the tropical paradise of Ohio all the way up to North Dakota. But in the future, that would be an area where we would want to have more partnerships because the Northern Great Plains is so important for ash tree protection over time, especially because it's one of the only hardwood trees that'll live up there. So it's really, it's a great question.
Right.
And Connor, I would encourage you to reach out to Jennifer or Rachel or myself if you prefer, to see how we can potentially get you the information you really need in a more formal fashion. So. And with that great note of sharing resources, I think we are going to officially call this webinar to a close on the hour. Please remember to register for additional webinars throughout the week. They are on all sorts of different topics. And thank you to our presenters and everybody who participated in the Q&A. We really appreciate you all.
Yeah, thank you. Have a good day.
Thanks everyone.
You too.